I'm recording this now. Okay, so we're recording. First of all, welcome to the first PGC Worldwide Lab Meeting that we've had this year. As you know, we're trying a new system. We're using Zoom instead of the old school approach to this. By and large, we've been pleased with how Zoom has worked, but this is the first time we've tried the webinar function. So we'll give it a go and see how things work. It's a distinct pleasure to introduce to you Gerome Breen and Helena Gaspar, who will be giving the talk today. I think Gerome is well known to all of us, and I've been particularly impressed with this piece of work, which really has done a really interesting and careful job of looking at the schizophrenia GWAS findings vis-a-vis a completely independent set of antipsychotic targets. It's a really interesting piece of work, and I think it actually is extremely consistent with the PGC's new mission, which is to try and find actionable variation and to see how these results can actually inform drug discovery. So at this point, I will turn this over to Gerome, and Gerome, please go ahead. And it looks like we've got 52 people on the call, so this is actually awesome. So Gerome, over to you, please. Gerome, your microphone is muted.
Okay, I'm unmuted now. Great. Okay,
now we can hear you, and I can see your slides too.
Great. Okay, so thank you, Pat. So we're going to talk today about how we might go about translating psychiatric genetics, and specifically about how we can use biological pathway analysis to potentially find new drug targets and new drug repositioning opportunities.
Gerome, can I step in for one moment, please? Yeah. Everyone on the call, now there's 55 people, that's fantastic. Listen, if you want to ask a question, there are two ways to do so. Down at the bottom of the Zoom screen, there's a Q&A panel, and you can ask a question through that. There's also a chat function, and you can ask a question that way. So please go ahead and feel free to do that throughout the call. Sorry, Gerome, back to you.
Okay, thank you. Just to take a step back for a moment, so we're all kind of very familiar with GWAS, but it's often forgotten exactly how recent GWAS is and exactly how much of a difficult start it had. So I think you could say, like all new technologies, GWAS had a peak of inflated expectations around 2006. Well, what's remarkable about GWAS, however, was when the trough of disillusionment was encountered, which was something around 2008. However, we can now say that with what has really been an astonishing wave of discovery in GWAS for psychiatric disorders, but also for every other sort of set of complex disorders, that we've now reached a stage where, at least for the discovery of GWAS hits, we know what we need to do. We have the mature technology, and in Gartner curve terminology, we've reached the plateau of productivity for GWAS hit discovery. But in terms of GWAS, we've often focused on how many GWAS samples do we need in order to make a discovery. And our current GWASs have given us a pretty good idea about the genetic architecture of psychiatric disorders. So classically, people have used, traits like irritable bowel syndrome, which has a relatively simple, high effect size genetic architecture, and height, which, although a continuous measure, is actually very similar to human complex disorders in that it has a highly polygenic, low effect size architecture. And here we just plotted, here is plotted the number of thousands of cases that you need to obtain a given number of genome-wide significant hits. You can see that IBD has nearly 100 discoveries for about 6,500 cases. And height, however, took nearly 40,000 cases to get the same discovery rate. Schizophrenia, it appears, is slightly more efficiently GWASed than height is. So it's got a slightly more simple genetic architecture, higher effect sizes. But bipolar disorder, for example, possibly has a more complex genetic architecture. Now, other disorders like depression have an even more complex genetic architecture than bipolar, but they're also beginning to yield hits. Now, what does this mean in practical terms? So what is the endpoint of GWAS? So of course, we've got GWAS hit discovery. Now this figure here comes from the supplementary material from the Ripke et al. PGC 1 plus Sweden analysis. And it's really quite well worthwhile reading the supplementary material in this paper because of the Bayesian modeling that they did to predict from the observed effect size distributions how many SNPs that might potentially underlie the observed heritability for schizophrenia. Now they concluded that something around 8 to 12,000 SNPs might underlie, or independent loci might underlie schizophrenia. They also calculated how many cases you might need for a given discovery rate, and they made some projections. Now, these projections are more or less accurate. It has to be said, however, that we're probably at the lower expectation of the projection here, so that we're on the green curve for discovery for psychiatric disorders. But if I move on to what are the practical implications, so this modeling suggests that there may be more than 5,000 or even more than 10,000 loci for pretty much every psychiatric disorder. You can work out based on this modeling that you probably need to genotype maybe more than 250,000 schizophrenia cases to identify most variants for schizophrenia. For other psychiatric disorders, you probably need to do larger sample sizes, so perhaps more than 1 million or 1.5 million for MDD. It must be said, though, for MDD, because MDD is present in significant numbers in almost every large genotype cohort and biobank, that will probably reach 1 million for MDD in the next decade or so. But much before we exhaust discovery for psychiatric GWAS, we should begin to tag genes and pathways. We should begin to generate powerful polygenic risk scores for not just schizophrenia, but every other psychiatric disorder. But it's the molecular pathways which I want to talk about today. And this speaks to how we can translate genetic findings. So in the PGC, we fundamentally want to discover the genetic basis to psychiatric disorders because we want to understand the fundamental biology. We want to be able to inform clinical practice, and we want to be able to deliver new therapeutic targets to an at least partially stalled drug development pipeline. So biological pathway analysis. I think another point that can be made about that is that pathway analysis in genetic data is different to systems biology analysis using RNA and proteomic data, because those types of data give you general pathology. So this pathology is essentially the product of etiology plus the downstream effects of illness and treatment. However, in genetics, if you test pathways for association using genetic data, you aim to understand etiopathogenesis. And this can help us to develop a better understanding of biological etiology, the underlying causes of the illness. But why is this of use? It's use because ultimately, if we can understand the causes of psychiatric disorders, we can develop better treatments for them. Now, last year, I convened a writing group coming out of the writing of the PGC 3 grant. One of the aims was that we get some review articles out about our aims. And I convened a writing group bringing together PGC PGC investigators and industry investigators to tackle the topic of how we might go about translating GWAS findings. And it was an interesting series of discussions that we had, and we published a review article in Nature Neuroscience last year. Basically, the sort of conclusions from that article and the group were that the predominant means in psychiatry and psychiatric genetics and in industry need challenging. So in clinical psychiatry, I think it's fair to say the impression is that drug development is very much stalled, and that all or most new drugs that are coming to market are based on previously discovered mechanisms and are Me Too drugs. In industry, they perceive that there's a lack of new targets coming out of psychiatric research. And there's also the perception that pharma is not investing in psychiatry. Now, as means go, they're all perhaps partially right, but actually mostly wrong. So, for example, there are 183 trials currently ongoing in PGC disorders in industry. There's actually billions of pounds of investment still in research and trials in the pharma industry. It's just that that's I think what we actually see in psychiatry is that the investment in psychiatry, of course, is less than the investment in cancer, for example, but it's still very, very considerable. Likewise, there are lots of new targets emerging from the genetic studies of psychiatric disorders, and we need to make pharma more aware of these. Now, one of the problems with looking at GWAS data is that the existing drug discovery toolbox is geared to rare mutations. GWAS hits are quite different to rare mutations. They often don't have good indications as to their functionality, and you really don't have a good understanding of the direction of effect in terms of the functional impact of a GWAS hit until much after a GWAS hit has been discovered and a lot of functional work has been done. But a general understanding of the additional steps required to identify a drug target has developed. So people know roughly how to do GWAS locus to gene mapping via studies of the function of disease or disorder associated SNPs. And some therapeutics targeting GWAS identified targets, such as HM-GCR, have been successfully developed. GWAS hits, we are developing a better understanding of them. We do know that GWAS hits are loci, not genes, that a GWAS locus often, but not always, includes multiple genes within the region of statistical significance. What has happened in the last few years is that expression, protein, and methylation QTL mapping approaches have been able to be used to try and better map causal genes. It has to be said that, with the exception of a few disorders, that there are few large samples of available brain tissues from both patients and healthy donors. Some people are working hard to change this, particularly for schizophrenia and bipolar disorder. But, for example, there are very few OCD and ADHD brain collections. Now, an absolutely superb resource is the Common Mind Consortium. So this is a consortium studying, I think, up to 1,000 brains from schizophrenia and bipolar and controls. And this is a paper, one of the first papers that they published in Nature Neuroscience last year, where they took data from almost 600 individual brains, multiple brain regions, and used this data to try and understand the functional impact of the schizophrenia-associated GWAS hits. They were able to suggest a functional role for maybe 20% of these hits, and were able to, with confidence, identify a role for 5 of the hits. Likewise, there have been other landmark publications in the last year. So the Seeker et al. Paper in Nature with Stephen McCarroll and colleagues, where they identified the C4-CNV that was underlying part of the HLA association in schizophrenia. They showed, how abnormal synaptic pruning may be important in schizophrenia, and currently that's driving some new therapeutic development. And there have been other papers such as Wan and Geschwind in Nature last year, where they use both statistical analysis methods and methods like HiC, looking at chromatin interactions, to demonstrate what the functional SNPs were in several GWAS loci for schizophrenia. However, these papers tend to focus on single GWAS hits, and each single GWAS hit requires a very large amount of work to actually clarify the functional role of the functional influence of the disorder-associated allele. And we want essentially to develop a method that allows us to go faster from GWAS hits to new drugs, to identify drug targets that are going to be relevant to these disorders. Now, parallel to the success in psychiatric genetics, the chemoinformatics world has been establishing large, very high quality databases that are cataloging drug molecule and small molecule protein interactions. This table shows various different databases. Kemble is perhaps the most famous of these, but there are also mental health focused databases such as KIDB, which actually serve a very valuable function to sort of counteract as it were, the cancer bias that you see in many of the public databases. But what we have here then is a potential treasure trove of drug-protein interaction information that if we can apply it to GWAS data, would actually be very valuable. Now, we concluded our paper by this figure, really, which was that if we can combine data from GWAS, which for psychiatric disorders will shortly include more than 1 million individuals, with this drug and small molecule information, we could then conduct drug target discovery. And we could identify not only potential single targets, but we could directly identify molecules that could be tested in cellular models and perhaps eventually in patients that have significant association with data. And if I go on then, so one of the aims or sub-aims of the PGC3 grant was to conduct this work. So we call this the Pathways to Drugs aim. And essentially, the idea is to take what has been a pretty straightforward biological pathway analysis of GWAS data and transform it to give us readouts that can drive rational drug discovery, especially discovery of drugs with new mechanisms in different psychiatric disorders. I say new mechanisms, but of course also the other thing that we can discover is drug repositioning opportunities for taking drugs that are approved in other disorders and seeing if they're indicated by the GWAS data. So I'll just skip that slide. So one thing to mention is a scale of pathways. So analyzing pathways is not necessarily a very straightforward business, because biological pathways come in all shapes and sizes, and you can have relatively straightforward and small pathways that have 5 or 8 members, but equally you can have pathways that have several thousand members. And one very good paper to read is this paper, The Statistical Properties of Gene Set Analysis, from Nature Reviews Genetics 2 years ago, where they compared different pathway analysis methods that exist for GWAS. But what they were able to show was that most of the existing methods had, to a greater or lesser extent, a bias against larger pathways, but also perhaps too high of a type 1 error rate. In particular, I would highlight that magenta here has a very, very high type 1 error rate, especially for larger pathways. But equally, some of the more commonly used methods also had a slightly too high type 1 error rate. The two methods that actually have very good type 1 error rates are Magma and Enrich. And as we're talking, as Helena will be talking about later, we mainly use Magma now for our analysis. So I'm going to hand over to Helena now to talk about our drug pathway analysis.
Thank you, Jerome. So I'm Helena, I'm not Jerome anymore. I'm going to explain how we do the pathway analysis for drugs. And I'm also going to talk about the pipeline we've been developing for the PGC to do the drug gene set analysis and the drug enrichment analysis. So first, some simple definitions. So what I consider a pathway is a collection of biologically related genes, and a drug pathway or drug gene sets is simply a set of genes with no interaction with the drug. So we can map a single drug to a single gene set, which is just a set of individual genes. For the moment, we do pathway analysis using Magma, so pathway analysis software, which assigns p-values to gene sets using SNP p-values from GWASs. So we only use, at the moment, GWAS summary statistics. We can also work from genotypes, but as usually we only have summary stats, it's easier and quicker to do it that way. So from SNP p-values, we can obtain gene p-values and also gene set p-values. Magma is a very easy to use software, and the gene p-values and the gene set p-values which are obtained with Magma are account for linkage disequilibrium. So for the gene set p-value, the null hypothesis is that the gene set is more associated with the phenotype than other gene sets. It's a kind of competitive analysis. For the PGC pipeline, we wanted to take the GWAS p-values from the PGC and be able to rank genes and drug targets, and also rank pathways and drugs, and do some kind of drug class enrichment analysis. We also want to do the opposite, take any set of genes and rank the PGC phenotypes. So again, for this, at the moment, we use Magma for the gene set analysis. As for the data sources, at the moment, the pipeline integrates several pathway sets. We have biological pathways from NCDB, drugs from the drug gene interaction database and the KI database, diseases from open targets, and, well, simply gene families from the HUGO nomenclature website. We are integrating more data at the moment. We want to get the common mind data and so on. And if you have interesting pathway sets, please do tell. We'll be happy to process them. I'll go in a bit more detail about the drug databases that we've been using. The drug gene interaction database mines drug gene interactions from 15 different databases, such as Campbell, but also Drug Bank and Cancer Commons and so on. A lot of these drugs are cancer drugs, actually. We also mined data from the KI database. The KI is constant of inhibition. So this is a database from the Psychoactive Drug Screening Program, and it is more focused on psychoactive drugs. We actually wanted to enrich our drug gene sets with data from psychoactive drugs. From the drug gene interaction database, we got something like 11,000 drugs, but a lot of these drugs actually have exactly the same gene sets. So we've got less than 4,000 unique gene sets, and only 2,400 had more than one gene. By integrating the KI database, we had something like 300 more gene sets, which was a significant enrichment of drugs. So on average, 4 drugs have the same gene sets. And those gene sets only account for around 3,000 genes, which is approximately the size of the druggable genome. There is no precise size of this druggable genome. So this picture is from a Nature Reviews paper from Hopkins and et al. And the druggable genome is approximately 3,000 genes, so something like between 10 and 15% of the whole human genome. So I'm going to show some results that we got from the PGC2 schizophrenia. The figure that I have in front of me is composed of 4 panels. We've tested several pathway sets. We've tested gene families, we've tested drugs, and also biological pathways. The first panel, panel A, just shows the major targets of known antipsychotics. And as we already know, we have mainly dopamine receptors, serotonin receptors, adrenergic receptors, histamine receptors, cholinergic receptors. And then the other panels show the results that we got with pathway analysis. Panel B shows the druggable gene families. So just the gene targets which are in our drug set. So we had a significant enrichment for calcium voltage-gated channel subunits and C1Z domain containing, but the C1Z domain containing family mainly contains targets from the histocompatibility complex. For the top drugs, we got many significant top drugs shown in red, but they are mainly calcium channel blockers. I also show the top pathways in panel D. Here, nothing was significant for this analysis, but the top results show pathways related to synapses and voltage-gated calcium channels. I also show here a druggable genome Manhattan plot, because it's also interesting to look at individual drug targets and not only whole drug pathways. So it's just a usual Manhattan plot, except we only show genes instead of SNPs, and these genes are from the druggable genome. I also highlight families which are enriched, which are in the top results. So these include calcium channels, potassium channels, and those cholinergic receptors. So I've shown that we can see which are the significant targets, which are the significant drugs, but another kind of analysis which might be interesting is the drug class enrichment analysis. So not looking at one single drug, but several drugs, and see if we find those drugs of interest in our top results. To do drug class enrichment, we do a very simple analysis which uses an enrichment curve. So we just plot the percentage of non-drugs as a function of top drugs ordered by p-value. And for example, here I highlight a point where we retrieve 40% of the non-drugs in the top 500 drugs found by MAGMA. And you get a significant enrichment. The blue line shows the random enrichment, what you would expect if you had a completely random and null result. To assess this enrichment, we can use the area under the curve, and a random result would give an IUC of 0.5. A perfect enrichment would be IUC 1. To do the classification of drug classes, we just use the ITC classification, which is from the World Health Organization. There are 5 levels to that classification, and it encompasses something like 5,000 drugs. And I usually use a level 2, so something like 93 classes. For example, under the nervous system drugs, I only test for psycholeptics, coded as N05. Why do I do that? Because I want to have a sufficient number of drugs in my class to have some kind of power when I do the test. So we applied this method to schizophrenia, PGC2, and we got 2 significant enrichments for psycholeptics, N05, and antiepileptics, ATC code N03. I also show here the Q-value, so the FDR adjusted p-value, which comes from a simple Wilcoxon test. We also studied the effect of sample size on drug class enrichment. So the question was, do we get the same enrichment if we don't have sufficient power? If we increase the sample size, do we get better enrichment for the drugs of interest? So for schizophrenia, the drugs of interest are antipsychotics. They are the non-drugs. And I'm looking at the enrichment when I increase the sample size. So for PGC1, we didn't have any enrichment. Then for PGC1 plus Sweden, we got some kind of enrichment. And then for PGC2, we've got a very significant enrichment for antipsychotics. So this shows that we are actually able to see that antipsychotics are enriched. We retrieve them, they are the top drug class. So that might indicate that we can indeed use PGC2 GWAS to do some drug repositioning and try and find new drugs. But we couldn't do it for PGC1, for example. So this kind of validation is necessary if we want to do drug repositioning. We have to figure out if you have sufficient power, if our GWAS is valid to do this. Another side of our project, our PGC pipeline project for pathways, is to visualize these pathways. So there are many ways to do that. Why do we want to visualize them? Because when we just do the ranking, we don't really know how similar these pathways are, and how if they are really different or if they are really all the same but not that different. So we want to visualize it. So to do this, we can consider each pathway, each set of pathways as a pathway space, where each dimension is one gene. And the pathway will be described by several genes, so several dimensions. In order to visualize this, we have to use some kind of dimensionality reduction approach to generate 2D pathway maps, where each point is a pathway, and it can be colored by any phenotype. So we can use lots of approaches for this, but I prefer the generative topographic mapping because I'm familiar with it. And it's also a probabilistic approach. So it's really nice. We can work with probability density functions, and it's already been used for chemical space analysis. I'm just giving here an example of using generative topographic mapping to visualize pathways with this time bipolar disorder, not schizophrenia. So this is the GWAS study from 2012. This is a pathway map. Each point is a pathway, and I color by a phenotype. And here the phenotype is bipolar disorder. These are biological pathways. We didn't get anything significant here, but I only show the 3 first pathways. We have a cluster mainly related to regulation of muscle cell differentiation and a bit further away regulation of exocytosis, but they are not significant. This is another map, this time of diseases from the Open Targets website. They have something like 8,000 phenotypes and diseases. So at the top of the map, you can see that we retrieve what we expected, bipolar disorder and schizophrenia. The data that is used in Open Targets also includes GWAS hits. So we would expect to see bipolar disorder here. And we have also other clusters. There is one significant cluster which contains response to ziprasidone and rare female infertility, and it was very long, so I have not included the whole pathway name. And there is another cluster with cerebellar ataxia. So this is a drug map this time. So always the same thing, colored by bipolar disorder association. And so this time this is a drug. I've already explained what kind of data we're using for that. And if we look at the drugs, we can see clusters. Well, there is only one significant cluster, and it's calcium channel blockers with calcicebtin and pilipin. But we also have antipsychotics with cyclopentixol and flupentixol, and we have an antidepressant, citalopram. So these are only the 50 top drugs retrieved by Magma, and then I used a dimensionality reduction algorithm to give me this map, and I used the probability density functions to map a phenotype. So this is just an example of what we want to do, and we are going to integrate this in the pathway pipeline. At the moment, we have a very simple kind of output with PDFs and Excel files generated by Perl scripts. The PDF files include QQ plots, top pathway bar plots, Manhattan plots that I have already shown. Also, interactive HTML plots. For example, here for those biological pathways, you can just click on the pathways and the link will guide you to a page explaining what the pathway is. This is because of NZDB, because they provide links, which is actually very nice, so we can integrate this. So what is next? We want to integrate more pathways, of course. We're going to integrate co-mine, hopefully, brain and tissue-related pathways. We also want to enrich those drug gene sets. As I've shown, many drugs share exactly the same gene sets, so we want to enrich our database. So this is ongoing work, and we also want to do cross-trait analysis. And that's what I'm working on right now. And I'm going to let Gerome talk about our meeting, Pathway to Drugs.
Okay. Thank you, Helena.
Sorry, Gerome, I think there's been a question.
Okay. Okay, let me see if I can.
In the chat panel.
Yeah. Okay. Okay.
so so now when we went full screen, couldn't see that anymore. Okay, so you can ignore Pat and my banter in the chat screen. If I think we should use it.
Yeah, we don't know the direction of the drug effect, actually, as it is. Do they hear me?
I think so. Pat, is the questioner unmuted?
Yes, and I'm not 100% sure how to remute him. But the question is,
yeah,
I think it's clearly stated there.
Yeah,
and I think
so I think generally with MHC, right? So first of all, probably in schizophrenia, there's not just one MHC signal. And I think if you read the Seeker et al. Paper, they point that out. So the question, actually, I see here now, the question is only gone to panelists. Okay, so Pat, we're going to have to note this for the next time we run this. So basically, the question is saying, I think that for schizophrenia in the MHC region, we should just use mainly C4A for pathway and drug analysis. We should probably also exclude the large ITIH cluster in chromosome 3, as we do not know the exact causal ones. So question, drug pathways might be enriched. However, are we sure the drug effect is in the desired direction away from the disease state and towards normality or the other way around? Probably not. Okay, so there are two questions there, really. So one is just to say for MHC, there's not just one hit in MHC, probably. There's probably two or maybe more than that in schizophrenia. So we need to consider the region in general. We also tend to do our analyses excluding, with and without MHC to check if it's confounding things. The large ITIH cluster is actually in high LD, but actually there are quite a few other regions in the genome that are similar to that. And I think we've previously shown that pathway analysis can actually maybe map high LD regions. Then to get on to the second question about, are we sure about the drug effect being the desired direction? No, we're not. So genetic data is directionless. We can never see, we can't know for definite from genetic analysis whether you're finding a drug that causes, for example, depression, or will cure or treat depression, for example. But that's where we need to collaborate with neuroscientists and with industry, where they've got cellular models that can then be tested. We do still need model systems. We're not proposing that we can go straight from genetics to patients by any means. Okay, so I think does that answer the question? Yeah?
Yes.
Okay, good. All right, so then just to mention then about this meeting. So we've been working on this for a while, but of course, part of the PGC3 grant was to try and engage with industry. And with regard to this, we're having this meeting in London, March 2nd to 3rd, for PGC group leaders and representatives and the industry representatives. It's a day-and-a-half meeting held at the Institute of Psychiatry at King's College London, and we have actually an astonishingly good lineup of speakers. So I think you can see the program at this website if you want to go and look at it. If you're a member of a PGC group, there are a few tickets left, but write to your group leader and ask them to nominate you, and we'll be happy to consider or to send a few more tickets out.
Jerome, there's a new question in the Q&A panel.
Mm-hmm. Q&A panel, okay.
Yeah, that was what we were talking before.
So Helena, do you want to answer that question? Okay.
Yeah, I'm sorry. That's what I said about the druggable genome. So I'm going to say the question first. Do we stick to druggable genome definition that are from 10 years ago? Most gene products can now be drugged. drugs with different approaches, and our chemical weapons are expanded a lot. Yes, that's true. And I've said that the numbers that I've shown were not completely accurate, but the 3,000 genes number is actually what I got in my data by gathering data from the drug gene interaction database and the KI database. So this is actually what I got from the data. So, and we don't have a very precise number for this. And it's true that among those druggable genes, only a subset will be a disease modifier gene also, but we don't have precise numbers for that. So I'm sorry for that fuzziness.
All right. So I think I just wanted to close at one slide. So if I can try to come back on here. Okay. So I think this is my conclusion slide. So I think I just had some general conclusions that this work is promising, but actually to take this approach forward, we require substantial and integrated efforts involving the PGC, other academic groups, particularly neuroscientists and pharmacologists, and industry in a sort of pre-competitive framework. And essentially, we want to work collaboratively to drive forward target identification enough to begin clinical validation processes. And sharing of data and expertise will be essential. And then I'd just like to thank you for, these are my acknowledgements. We can take further questions. Okay.
There's a question in the Q&A panel.
That's about, yes, we'll be
interested to participate.
Okay, Sylvia, I'll ask. If you can put your email, send me your email. I'll email you after the meeting. Okay.
Great.
Okay. Other questions for Gerome? Okay, so I'll ask one. So I guess the one thing that was really striking about this was how much better things got as you were going from PGC 1 to 2, or from the earlier to the later stuff. Have you guys done any power calcs or so how much better do you reckon things will get if one were to look at the CLAS UK results, or if you were to get up to the approximately 60,000 cases that the PGC schizophrenia group is thinking they're going to get in a couple of months? Any thoughts on that?
I don't know. I haven't made any special calculations, but I think there will be a plateau.
There will be a plateau after a while. However, we can say that on the early freeze of the PGC3 schizophrenia data, which I think which Stefan presented in Jerusalem, Helena ran this pipeline, and in terms of biological pathway identification, it was actually performing noticeably better already. So it does appear that I think we've known this for a while, that biological pathway analysis is not a magic bullet. It won't take a negative GWAS and solve it for you, but it will help you make proper sense of a large and very successful GWAS.
Makes sense. Is there anything else that one could do to tweak this a little bit? I mean, would it be sensible? I mean, if you were to get a list of genes that were never expressed at all in any study in brain, for example, would that help you? So in other words, if you were to really focus on genes where we've got excellent evidence that they're not part of the CNS, and obviously under the assumption that schizophrenia, et cetera, are brain diseases, would that palpably help this or not?
No genes.
Well, genes that are not expressed in the brain. So I think the answer is yes, that all of those things can help. However, you have to bear in mind that there's a quite sparse information about the expression of genes in critical developmental risk periods for most psychiatric disorders. So, for example, the number of brains that we have from early adolescence
is very small, and equally, brains from ages 0 to 10, those are relatively few in number, and I think that gives me some pause in terms of implementing gene expression filters on the data.
Okay, thank you. And there's a question from Marty Farrell.
When you use the KI database, do you use all of the drug targets, the full range of KI values, or do you have a KI cutoff? Well, of course I have a KI cutoff because I'm only selecting what I consider the active interactions. So the KI cutoff, if I remember well what I've done, it was between 7 and less than 14.
Thank you.
In fact, they are reduced to binary interactions. We say, oh, it's active or it's not active. And I say if the cutoff is respected, well, it's active. Otherwise, I don't take it into account.
Okay, are there any other questions for Gerome or Helena? Okay, I don't see any other ones popping up. Okay, so it looks like we peaked out at about 75 people on this call today, so that's actually excellent turnout for this. So thank you very much for the fantastic presentation. And I guess the one final thing I wanted to ask Gerome is, do you guys have any plans to live stream the conference in London, or to make it available in some way to people offline?
Yes, we're considering whether we can record and make it available.
Okay.
It's the first time we're running it, however, so we haven't addressed all of these issues.
Yep, fair enough.
Okay.
Okay, well, listen, I think a fantastic talk, and I really appreciate you guys taking the time to do this, and it'll be fun to see how this work progresses with larger and larger sample sizes, and also with other disorders as they come into play. So I'm sure people will join me in thanking you for taking the time to give such a great talk today. Gerome and Helena, thank you very much.
Thank you. Thank you all for listening.
And that will close this worldwide call. So everyone, thank you very much for your attention today.